contraction is how calcium, which is stored in the sarcoplasmic reticulum, terminal cisternae of the sarcoplasmic reticulum, is released. And when it's released, it binds with troponin, which is on the actin molecule, causes the troponin to change shape. And when it changes shape, it moves the tropomyosin out of the way, and you get binding of the myosin head to the actin, and then the filaments slide across one another. You saw that in the video, and it's pretty easy to understand when you see it in a nice video like that. It's so difficult to explain, you know, verbally, but when you see a video of it, it's like, oh yeah, I get it. Calcium, movement of molecules, binding of heads, and the sliding of the filaments. So anyway, so this is the idea of what the sliding filament is all about. Now, the key, of course, as we've mentioned many times in here, is calcium. And so calcium is stored. Let me get this model. I know everybody's probably looked at this model. Let me get it anyway. So here's a muscle cell. And on the muscle cell, of course, these little blue lines here are the T-tubules, and that's where the action potential penetrates into the muscle. And we were just talking the other night, I was talking to somebody the other night, asking questions about this. So this kind of webby looking, kind of, I don't know, brownish beige stuff that kind of covers, you know, the sarcomere here, that's the sarcoplasmic reticulum, the SR. The terminal cisternae just refers, there's a picture on the slide just a second, refers to this sort of kind of little fatter section of it, which is just adjacent to the T-tubule. So it makes sense if you think about it. I mean, you know, the action potential comes in the T-tubule and stimulates the calcium ions to be released from this sort of little, you know, bigger, fatter area of the sarcoplasmic reticulum. And then you know what happens next. It interacts with the troponin and the tropomyosin and all that sort of stuff. So basically what happens, you know, is the action potential goes through the T-tubules, stimulates release of calcium at the terminal cisternae of the sarcoplasmic reticulum. All the conformational changes occur, and the sliding filaments, you know, the filaments slide. Okay, so we'll do this picture. So you can see in the background here, this is sort of just the opposite of our model, you know, the colors. But anyway, so the blue thing here, of course, is the sarcoplasmic reticulum. You can see that it's covering, loosely sort of covering the sarcomere. And, you know, there'll be another sarcomere here, and, you know, so on and so forth. So here's the T-tubule. These little expanded regions here are the terminal cisternae. It's the terminal cisternae right here. And there's another word, I don't think it's in my slides, and I don't think it's particularly that keen to be that we actually use this word, but this word here, triad. What the word triad is referring to, tri meaning three, is referring to, like, you know, terminal cisternae, T-tubule, terminal cisternae, kind of a group of three, and so they call that a triad. Just a little bit more terminology that you need to, you know, commit to memory. So this is the idea, the relationship between the T-tubules, which are going to propagate the action potential, and very nicely with the terminal cisternae where the calcium is stored. So that's pretty cool how that's all works together. Yeah.
So the triad where the terminal cisternae are, all of that will always be the overlap area.
Yeah, it's just the overlap. Yeah, exactly. Yeah, what they said, triad, and it's right at the zone of overlap. So, you know, and that's what's going to start our contraction.
So where it ends is where the A line. And
that's right. Yeah.
And then going on inside is the
yeah. The
I forget it. H zone.
H zone.
Yeah.
No. Well, I think I have another picture to show too. T-tubules, let me see. Okay, so we can do this one. It's a busy slide. But anyway, the T-tubules distribute. That's the word I was looking for. God, I just couldn't remember that word. Distribute the electrical signal. You know, I called an electrical signal. A physiologist would slap me, you know, upside the head and go, it's not an electrical signal, it's an action potential. But you know what? I'm not a physiologist. This is an anatomy class, so they're going to have to get out of here. Anyway, so they distribute the electrical signal into the muscle fiber. And you know, then the calcium ions are released, interaction with troponin that moves, changing position of the tropomyosin, and we get contraction. So I think we beat this thing to death now. Okay, you get the idea that calcium is important. and how it interacts with the tropomyosin and in turn with the troponin and the tropomyosin. So that's the idea of the T-tubules. They're the distribution channels. There. I knew there was an easy way to explain it. All right. So, okay, so a couple of things here. Here's our neurotransmitter, the acetylcholine, which is stored in the synaptic terminal. You've already had the nervous system. You can't have forgotten it already. Well, you could have. But anyway, so remember at the end of the axon, so my arm is an axon, and my fingers are the synaptic terminals, and then the tips of my fingers are where the acetylcholine is stored in the little vesicles. So when the action potential, this of course is a motor neuron because it's going to muscles, right? So the acetylcholine is released. Lots of other things that we don't need to deal with happen, and we basically get muscle contraction. The word motor unit is defined in your book as all the muscle fibers controlled by a single motor neuron. Now remember, from the nervous system, the motor neurons go out to our skeletal muscles, they deliver an impulse, and it tells the muscle to contract. So what would happen, let's say, with like nerve damage or something like that? Let's say that this little fiber somehow was destroyed or, I don't know, damaged or something like that. Well, then you might have loss, perhaps only temporary, but some loss of, you probably wouldn't even notice it because it would be so small, but maybe some loss of motor fibers in a certain area. Because if the impulse can't reach those fibers, then you're not going to get contraction. But there's usually enough overlap that probably won't be the issue. So a motor unit, all the fibers, and there may not be that many that are controlled by one motor neuron. Or I guess you know what? I just thought it. Maybe in an extreme case, if we look at something like ALS, Lou Gehrig's disease, where the motor neurons function but their pathways are destroyed, then you can see in the extreme case, nothing happens. You lose all voluntary muscle control, and then perhaps ultimately die from, the suffocation or something like that. Okay, so this is the contraction idea, and acetylcholine is the one you got to remember. Let's see here. This works. There's a couple of things here. I guess the thing to look at, maybe this little box right here. So here's our synaptic vesicles. This is at the end of my fingers. They contain acetylcholine. When the action potential arrives, the acetylcholine is released, diffuses across this extremely tiny space, binds with receptors on the surface of the skeletal muscle, and the action potential then is distributed into the muscle. Also, we find in this little cleft here acetylcholinesterase molecules, which are an enzyme which breaks down the acetylcholine, and the cycle will then be interrupted or not continue because the acetylcholine is being broken down. So this would be this thing called the neuromuscular junction. You've seen a slide of this before, and I'll show it to you again, the junction between the nervous system and the muscle. Another picture of it, this is maybe more familiar with the one we saw earlier in the semester. So here's our nerve, here's our little fibers, and here's our synapse between, you can see the skeletal muscle, and this is where the little synaptic vesicles are located where the acetylcholine will be released from. So that's pretty simple. And then these are two pictures here. That's the last one. This is the first one. These are from your book, which deal with the cycle of contraction and relaxation. Now I know, as I've said many times, this isn't a physiology class, but we have to do a teeny bit of it. So here's our little motor interplay. Here's our synaptic terminal. This is where everything is going to happen. So number one, acetylcholine is released, and it's bound to receptors on the surface of the muscles. Then we get the release of the calcium ions from the sarcoplasmic reticulum, which then, of course, we get all this business about the troponin and the tropomyosin. Cross bridges are formed between the actin and myosin, and the filament slide. Right? So that's the contraction step. The relaxation step, see right up here, initiating muscle contraction. The relaxation steps, acetylcholine is removed by acetylcholinesterase, which is the enzyme which breaks it down. You get all this stuff in physiology, I promise. The calcium is reabsorbed by the sarcoplasmic reticulum. The molecules, the troponin and the tropomyosin, then return to their original conformation, covering the binding sites. and relaxation occurs, because the myosin can't bind and the muscles relax.